Hi, everyone. I'm so excited to have you all join us today. Welcome to the third webinar of this year's AGM Preclinical Translational Science webinar series. For who that doesn't know me, I am Serene Markosian. I lead the Assay Guidance Manual program here at the National Center for Advancing Translational Sciences, which is part of the National Institutes of Health. We are absolutely excited to have an amazing speaker with us today. So I'm really excited to introduce Dr. Raymond Deshaies as our speaker. Dr. Deshaies directed basic and applied research programs for over 30 years as a professor and Howard Hughes Investigator at Caltech, and later as the head of global research at Amgen. While at Caltech, he co-conceived and developed, this is with Craig Cruz at Yale, proof of principle for targeted protein degradation with PROTACs, and co-founded two companies, one of which developed Kyprolis carfilzomib. His fundamental research at Caltech was recognized by election to the U.S. National Academy of Sciences in 2016, and his applied research with Dr. Cruz has been honored by the Gabe Award in 2023 and Passano Award in 2025. While leading global research at Amgen, his team advanced nearly 5 dozen clinical candidates, including Lumacras, sotorasib, and others. We're super excited to have him with us today. And before I give him the floor, I'd like to mention that if you have any questions for Ray, please drop them in the Q&A box and we'll get to them at the end of this presentation. Ray, on behalf of us, NCATS, all of my colleagues here at NCATS, all of our attendees, we're really excited to have you here. We're excited to hear your talk, and the floor is yours.
Thank you so much for the introduction, and thank you so much for the invitation. It's really a privilege to have the opportunity to come in front of all of you today and talk a little bit of science. So without any further ado, the theme of my talk is that industry is really in the midst of a major transition in the type of molecules that we make. And this is something I first pointed out in a review I published in 2020, in which I argued that there have been 3 transformative waves in the history of the industry going back to its founding about 125 years ago. The first was making specific defined molecules as drugs. The second was the advent of rational drug design, where instead of using pharmacology as the tool of finding drugs, picking targets biochemically and then making molecules that directly interdicted those targets. That was rapidly followed by the biotechnology revolution, which gave us a whole new set of modalities related to biologic molecules made inside of cells, and has really transformed the type of therapies that are available. And we're now in this early stage of this 4th revolution of multi-specific medicines. And these are medicines that differ in a really important fundamental way from everything that preceded them. So if you think about, the prior medicines that have been in development for the past 125 years, they are by and large monospecific in that you have a target and you have a drug, and you form a drug-target interface, and you have a single interface that then either drives up the activity of the target or diminishes the activity of the target. And that's true for aspirin bound in the active site of cyclooxygenase 1. It's true for a natural protein like erythropoietin binding to its receptor and agonizing it, turning it on. And it's true for an engineered monoclonal antibody therapy like Vectibix panitumumab that binds to the EGF receptor and blocks the binding of epidermal growth factor to antagonize signaling through the receptor. Now, the distinction is a multi-specific can contact 2 or more things at the same time. And what I'd like to argue to you is that's not just a difference in quantity, that's a difference in kind, right? So it's not like you're twice as good because you combine 2 things. You can literally do things that you can't do otherwise with a monospecific. And so therein, I'm now going to describe what I think are 2 sort of major advantages of multispecificity, which is really driving the field in that direction. The first I've just alluded to, which biology imposes critical barriers to pharmacotherapy, and that limits what you can achieve with the traditional monospecific drugs. Multispecifics enable you to surmount those barriers. Now, what are those barriers? Well, the four that I like to talk about or think about are illustrated on this slide that I'll work through. The first is stability. A lot of molecules in the body are just cleared very rapidly, so rapidly that they could not be practically used as therapeutics. And one strategy for dealing with this is to make a multispecific where one part of the drug is carrying out the pharmacology and the other part enables recycling of the drug so it has longer persistence in the body. And the first example of this was actually an Amgen drug, etanercept Enbrel, which is a TNF receptor ectodomain fused to the Fc domain of an antibody. The ectodomain by itself is very unstable. It could never be a drug. When you fuse it to the Fc domain, you get a dramatic stabilization of the ectodomain of TNF receptor, and now have a molecule that can essentially serve as a sponge to stably soak up TNF receptor. And that drug Enbrel has been very successful. And there's many other examples now of Fc fusions, albumin fusions, albumin binding molecules that have this recycling capability. Another thing that's a limitation posed by biology is redundancy. Now, many different biological outputs can be controlled, have multiple different pathways impinging on the output, such that if you block one pathway, you don't really change the output necessarily. You have to block multiple pathways. And we can see this intuitively in oncology or virology, where increasingly a lot of the drugs are actually combinations that are attacking multiple pathways at the same time. With a multi-active multispecific, as shown here with a bispecific antibody, you can simultaneously attack two different pathways, for example, that might be sustaining cancer growth. Amivantamab from J&J is an example where it blocks EGF receptor plus a major bypass pathway, the MET receptor. A third limitation posed by biology relates to therapeutic index. And often the conversation around therapeutic index has to do with specificity of a drug, especially a small molecule. And if it hits an unintended target, it could have a low therapeutic index. But even if you have perfect specificity, 100% specificity for your target, you will still have a problem with therapeutic index. And that's because during evolution, our genomes have become very efficient repurposers of genetic information. And that's the simple consequence of the fact that we have a very restricted number of parts, about 20,000 different genes, compared to, say, a 777 Boeing, which has literally over 120,000 unique different parts in it. So we have many fewer parts, yet are incredibly complex. The way this works, essentially, is that the same parts get reused again and again and again in different tissues at different times of development to do either the same, a similar, or sometimes quite a different task. And so even if you have perfect specificity, for example, you block mitosis in a cancer cell, the reality is normal cells that have to proliferate to replace your blood, to replace your skin, also have to go through mitosis using the same components, and that leads to on-target side effects. With a multi-specific, you can attach a zip code, if you will, to the drug, an address label, to deliver it to exactly where you want it to go, as in the case for the siRNA shown here, targeted to the liver, or antibody drug conjugates, for example, targeted to cancer cell antigens. Finally, the final major limitation posed by biology is druggability. Whereas we know as an industry how to drug cell surface receptors with defined ligands, we know how to drug many different types of enzymes, there are certain types of factors, proteins, that we really don't know how to drug all that well. Transcription factors being a dramatic example of that. Many proteins are unstructured, they don't necessarily have active sites, or pockets that small molecules can bind. However, we know that cells don't face this limitation. Take transcription factors again. We know that they are potently regulated by ubiquitination, by sumylation, by phosphorylation, by acetylation, by tethering to the cytoplasm. And so the idea here is the drug itself doesn't carry out the primary pharmacology. The drug is a matchmaker that links your target to a natural effector mechanism that pre-exists in the body. And so you're essentially bootstrapping on what already exists in the body to carry out the pharmacological action. Now, there's another reason that's really driving this ascendance of multispecific drugs, and that's that there's a paucity of high-quality novel targets that is driving industry to intensely focus on a small set of validated targets. And this was beautifully illustrated in a review by Fournier et al. In Nature Reviews Drug Development a couple of years ago. I've stolen this graph from them. And it's worth cogitating on this graph for a bit because the implications are just so important. What they show are the top 25 targets that industry has worked on the x-axis, and they graph the competitive intensity on the y-axis in 2000, in 2010 in dark blue, and in 2020 in purple. And what you can see is that competitive intensity has gone through the roof. For the 25th most popular target, it's roughly tripled since between 2000 and 2020. And I would wager that has gone up even further in the past 5 years. And what you see for the top target, it's gone up more than fivefold. You now have over 50 programs on the most popular targets. And so the likelihood that any one company can succeed in that kind of environment is very slim. What multispecificity can do is it allows you to reload against the same targets that have been successful in the past. But by bringing in multispecificity, you can now do things with that target that you could not do with a monospecific. And I think the ADC and HER2 is a great example where now you can treat women with breast cancer with levels of HER2 expression that are not addressable with trastuzumab, which is the conventional Herceptin monoclonal antibody. So you really open up a new window that allows you to reload using known existing targets to achieve effects that you could not achieve otherwise. But mind you, you can also go after new targets as well. So I think these are two reasons. You can do stuff you couldn't do otherwise, and it opens up the window and gets you out of this insane competitive race with monospecific drugs. Okay, now the net effect of this is there's been a real explosion in the growth of these drugs. I've been tracking prospectively developed monospecific drugs over time. The first was Enbrel, which I've already mentioned in 1998. That's an FC fusion. And what we've seen is that there's a rising slope of these approved prospectively developed drugs. From having no approvals prior to 1998, in the 2022 to 2024 window, 1 in 6 FDA approvals was for a multi-specific drug. And half of all the approvals of these drugs that are graphed here have occurred since 2020, since the pandemic, since I wrote that review. So given the long latency of product development cycles in this industry, that shows you how rapidly this is going. But even more impressive is look at this. These are trials of multispecifics initiated per year. Note that the y-axis here on the right is tenfold greater than the y-axis on the left. What you can see is that this rise is even steeper than the rise of approvals. And so there's an enormous amount in the pipeline. So I wager that we will see an increasingly greater share of new drug approvals going out to multispecific molecules. Now, I'd like to focus for the rest of my talk on a particular class of multispecific molecules. And these are what are referred to by a variety of terms, but here I'm going to use proximity medicines. And I'll start talking about induced proximity. And these are matchmakers. I've already talked about matchmakers earlier on, where I showed the 4 different advantages of multispecificity. So these are molecules that contact an effector and the target simultaneously, and that allows the molecule to induce the effector to act on the target. And there's all sorts of things you can do with this. The most popular right now, and I'll talk about that on the next slides, is targeted protein degradation, but you could induce other covalent modifications, demodifications like phosphatase, removing phosphate, or even relocalizing a protein to a location where it'll have more or less activity. And importantly, this mechanism is not limited to inside the cell or outside the cell. You can use induced proximity pretty much anywhere that proteins reside. And it's not limited to modality. You can do induced proximity with small molecules, with biologics, with complex synthetics like peptides, or even hybrid modalities. So this is finally, you're not even limited to molecules. You can use induced proximity with cells. And in fact, that's where most of the initial progress has been made. So this is just a list of the induced proximity molecules that have been approved by the FDA to date. These are all prospectively developed induced proximity molecules. And as you can see, almost all of them, starting with catamoximab, are CD3 engagers. So these are T cell engagers that bind a T cell and they bind a cancer cell. They switch the T cell on, and by inducing the proximity of a T cell and a cancer cell, they induce the T cell to kill the cancer cell. And these account for most of the induced proximity approvals to date, although I think we're going to see the breadth of opportunities here, or the breadth of options, the breadth of molecules is going to increase dramatically if you just look at what's in clinical development right now. But this shows this is a very broad category of mechanism. Now, I'm going to talk about induced proximity for targeted protein degradation, which makes use of the ubiquitin system, a natural system that exists in the cytosol and nucleus of cells. Now, let me give you a little bit of a primer on the ubiquitin system. Ubiquitin is a small protein, and it gets physically conjugated to other proteins in a covalent linkage. And for that to happen, the ubiquitin needs to be activated by an enzyme known as E1, ubiquitin activating enzyme. It's transferred to an enzyme known as E2, ubiquitin conjugating enzyme. And then the E2 joins forces with an E3 known as ubiquitin ligase, and that complex then transfers ubiquitin onto a target protein. Now, there's multiple possible outcomes of ubiquitin attachment. It could be a signaling motif like phosphorylation, but a common output of ubiquitination is that the ubiquitin signal is recognized by the proteasome, a cellular protease complex, which then degrades the ubiquitin conjugate. Now, in parentheses here are the number of genes encoded in the genome for these various functions. The one to note is E3's, ubiquitin ligase, is over 500. The number is somewhere 500, 600, a lot of them. That's on par with protein kinases. And the rationale there is these are the enzymes that, for the most part, confer substrate recognition as well as regulation. Okay, so by having a lot of them, it gives the system enormous plasticity to recognize many different substrates and to regulate those recognition events. Now, ubiquitin plays 2 very general roles, ubiquitin degradation in the cell. One is proteome-wide, which is that proteins are constantly misfolding or they're getting damaged by environmental toxins like oxygen. And those proteins need to be eliminated. And ubiquitin system is deployed for that. When humans inherit mutations or acquire mutations that disable this system, they are prone to disease, including neurodegeneration. The other way the system is used is to degrade proteins that are perfectly normal and active, but that you want to remove from the cell to achieve a regulatory purpose. And the first example of this was cyclin, where you need to degrade mitotic cyclin to exit mitosis so you can shut off cyclin-dependent kinase activity. But we now know there are many, many hundreds, if not thousands, of examples where degradation is deployed in a regulatory capacity. When humans inherit or acquire mutations that disable these specific regulatory functions, they are prone to a number of different diseases, including prominently cancer, where one of the most commonly mutated tumor suppressor genes, CDC4, is a ubiquitin ligase subunit, also known as FBW7. Now, one of the things that really got me to thinking about the therapeutic possibilities of deploying the ubiquitin system was this remarkable paper that Peter Hawley published in 1993. He was studying human papillomavirus, in particular the E6 protein, which is critical for the viral replication cycle. And he and his students found that E6 binds a protein in the cell they called E6 associated protein, E6AP, which is a ubiquitin ligase. E6 also binds P53. And so it was effectively inducing the proximity of E6AP and P53. This drove rapid ubiquitination of P53 and its elimination from cells. Now, P53 is normally quite unstable, but when E6 is present, bringing in E6AP, it is so unstable that it is no longer detectable. And this elimination of P53 essentially got rid of a factor in cells that restricted the replication life cycle of human papillomavirus. So this served the virus's ends. Now, this is not a one-off observation. Others followed up on Hawley's observation, and this is an incomplete list I have on the right of examples that have been found over the years. And you could see many different viruses employed this strategy to go after multiple different targets inside of cells that would normally act to restrict viral replication life cycle. Now, the thing that really strikes me is viruses are masters of genomic economy. Take HIV. I think the genome is somewhere around 5 kilobases. Every single nucleotide is parsed out for its relevance and eliminated if it's not useful. The viruses have to maintain the smallest, most compact genome. Now, HIV, despite that, deploys 4 different small ORFs, each of which eliminates a different cellular protein that would otherwise restrict the viral life cycle. So if viruses have landed on this mechanism and used it many times, that argued to me this is an extremely powerful and effective mechanism for taking over the physiology of a cell. And if viruses could do it, perhaps we could as humans as well. And this led Craig Cruz and I to conceive of this idea of making molecules that we called PROTACs, where one part of the molecule brings in a ubiquitin ligase. I'm sorry this is such a complicated drawing. This is like from our original 2001 paper, where I was all, my head was in the details of Cullen ring ubiquitin ligases. So this is an SCF ubiquitin ligase. On the right, it's bringing in a target, a protein Craig's lab had a deorphanized an inhibitor for. The target is methionine aminopeptidase 2. By linking these two, the ligase to the target, you get ubiquitination of the target and degradation by the proteasome. So that was the cartoon drawing. We actually could show that this worked in vitro. When we mixed everything together, that's lane 4 here. You see this high molecular weight smear that's characteristic of ubiquitination. If you use a chain terminating ubiquitin, you collapse the smear into a single discrete modified band down here in lane 2 and 3. If you leave out the PROTAC, that's lanes 5 through 7, or if you leave out the ligase, lane 1, you have no ubiquitination. So we could reconstitute ubiquitination using the PROTAC shown here in the middle. And that ubiquitination would productively lead to degradation, as shown on the bottom right, where we have a MED-AP2 and a frog egg extract. And this is a covalent modifier, this particular degrader molecule. The lower band is unmodified. It persists. The upper band disappears over time, and that disappearance is blunted by the proteasome inhibitors LLNL and epoximycin. So we could show in vitro and then inside of cells that you could repurpose a ubiquitin ligase to degrade a protein normally it would have nothing to do with. Now, over the next decade or so, there were multiple results that really cemented the potential of this. The first on the left, our result, but then Craig followed this up with many other demonstrations with increasingly sophisticated molecules showing that this was something you could do repeatedly and with molecules that would have drug-like properties. Next, at least in the way I think of the field, was this demonstration by Ning Zheng's lab at University of Washington that in fact, in normal biology, this exact idea is deployed. Plants use this to control one of their most important signaling pathways, the auxin pathway. Auxin works by initiating a transcriptional program. Normally, that program is under repression by these proteins, OXIAA proteins, that are potentially a substrate for a ubiquitin ligase, SCF-TER1, but the binding of these to TER1 is so weak that it's undetectable. And that's because there's a cavity in the binding site. Auxin fills that cavity, this little purple triangle, giving you now a composite surface that stabilizes the binding of the OXIAA proteins. They're rapidly ubiquitinated and degraded. So this was really incredible because it showed that, not only is this sort of a chemical biology trick, but nature has evolved to employ this mechanism to control one of the most important signaling pathways on Earth. Now, this is not just the example of Ning, because several years later, it was shown by 3 different laboratories that what was at the time the second best-selling drug in the world, Revlimid lenalidomide, used in multiple myeloma therapy, actually the best-selling cancer drug in the world at the time. However, the mechanism of action was not known. And what these labs shown, Ebert, Kalin, and Chopra, is that the way lenalidomide works is that it forms a bridge, essentially. It's a matchmaker that links 2 transcription factors, the IKZF proteins, to a CRL4-CRBN ubiquitin ligase. And what's remarkable about this is that these IKZF1 and 3, they're transcription factors essential for the survival of multiple myeloma cells. At that point in time, these were considered, transcription factors as a family, the most undruggable of the undruggables. Yet here you had the second best-selling drug in the world that was working precisely by drugging transcription factors. And so the confluence of all these results led to enormous interest in this field. And these are data that Beacon Intelligence helped me collect. There's been, right at this point in time, when we did this a year ago, there were 100 plus targeted protein degradation startups worldwide. There were 80 programs in clinical development. And for these 100 plus TPD startups, over 10 billion in venture capital had been raised. And that doesn't even count internal programs in established companies like Roche, like Amgen, like Novartis, and so forth. So enormous amount of enthusiasm and capital being dedicated to this approach. Now, here's a scorecard of where we stand as a field. As I said, 80 different programs. Almost all of those programs are targeting CRBN because what's been found is you could take lenalidomide and you can derivatize it with other molecules and very rapidly end up with a degrader that works via CRBN. And so many companies have taken advantage of that. So that's been really the dominant approach. There's a smattering of other ligases that have been used, but remember, there's over 500 of these. So there's enormous potential here to more fully exploit that biology. The number of targets, 35 different targets for the 80 programs. I think the important point here is that this is not a one-trick pony, right? This is not just IKZF proteins. You can deploy this mechanism for many, many, many different types of targets. Now, the molecule in the pole position in terms of development is vepdegestrant. This comes out of Craig Cruz's company, Arvinas, being co-developed with Pfizer. They released earlier this year data from a phase 3 clinical trial, and where they showed a substantial benefit in ESR1 mutated, ER positive, HER2 negative breast cancer. They have filed an NDA for FDA approval of that molecule. So we're all waiting with bated breath. This could be the first prospectively developed degrader to achieve an FDA approval. All right, so where's the field at now? Well, one important debate in the field is, should we be going after more PROTAC-like molecules or more molecular glue molecules? So what do I mean by that? Well, PROTACs are the molecules that Craig and I originally described. These are molecules which essentially have two different ligands, one for your ligase and one for your target, and then you join the ligands with a linker. And the advantage here is it's conceptually straightforward to develop these, right? You could find ligands, we know how to do that, and we know linker chemistry and join them and just, then optimize them. The challenge is that the medicinal chemistry tends to be very difficult because the molecules are complex, and you end up with molecules that violate multiple of Lipinski's rule of 5, so to speak, that are not particularly drug-like. The other approach, molecular glues, that's more like what auxin and lenalidomide are. They're very compact molecules. They tend to only bind to one of the proteins, and usually not even that tightly. They form a composite surface that the second protein can then bind to. And usually you see a lot of cooperativity here. The affinity goes way up when all three proteins are when both proteins are present, compared to the affinity for the one protein that it can bind. So these tend to be much more drug-like, much more compact. The problem is we don't really have great ways of finding these molecules. And so what's needed is something like more of a crank turning ability to find these molecules very systematically. Now, one approach that I've already hinted at, if you think of, I like to think of drug discovery as like finding a needle in a haystack. That's a typical drug discovery campaign, right? It's, you're having to sort through a lot of stuff to find your little nugget. If finding a normal drug is a needle in a haystack, then finding a molecular glue would be like a needle in a mountain, because now you're asking a molecule to do something really complicated, to recognize and bring together two different things, not just bind one thing. And so the strategy that I've alluded to for doing this is building bespoke libraries in which every member has part of the activity you want. So for example, if you take lenalidomide and you attach a lot of different moieties to it, you would end up with a library where every molecule can bind CRBN. And so the likelihood that you're going to bring in a substrate of interest to ubiquitinate and degrade it goes up dramatically, because every molecule potentially could do that, right? So that's one approach. The other approach is to figure out which ligase to pair with which target. And the thinking here is that these molecules tend to work best when the binding is cooperative. And it becomes cooperative because when you bring the target and the ligase together, they find some compatible surfaces that they can interact through that contribute to the overall positive binding energy. And it's sort of like what I show here with the puzzle pieces. It doesn't have to be a normal physiological match. It's just, that you fortuitously, when you bring them together, they have enough complementarity that you can get a little bit of binding energy there. And so there's a number of different strategies to try to find molecules that would be naturally sticky towards each other, even if they don't physiologically interact. So I'm going to finish the last part of my talk by going into an idea that I just published with Ryan Potts a week ago, last Thursday in Science, that touches on this question: what's next for the field of induced proximity, or for proximity drugs in general? So induced proximity is just like I said, you're inducing the formation of a complex. There's a lot of complexes that already exist inside of cells that are very unstable, that are ephemeral, right? The two proteins come together and they come apart very quickly. What if we can make things like this little wedge, which I'm going to call LOCKTACs here on, that would recognize these complexes and reduce their off rate and make them more stable and cause them to accumulate? And might those be interesting candidates for drugs? So I'm going to talk now about sustained proximity as opposed to induced proximity. So you could think of proximity drugs, as I've already alluded to on the slide I just showed, as being broken down into two categories. An induced proximity drug promotes a stable interaction between species that don't normally interact, or at least they don't interact in a way that has a notable physiological impact. And there's some argument around this point that, some induced proximity drugs, the protein may actually interact, like auxin with its repressor, the SCF-TER1 in plants with its repressors. There is a very weak interaction. This is very hard to detect. But we know there are examples where there's really no physiological interaction. Sustained proximity drugs, as I've said, there you have an interaction that naturally occurs already, and you're trying to stabilize it. And, what's cool about this in my mind is it's very general, and I'll allude to that in a coming slide. Now, if you think about all the drugs that have happened before, that have been developed in the past, I said, number one, most of them are monospecific, essentially almost all of them. Number two, they're almost all competitive inhibitors. And competitive inhibitors work through reducing the K-on of a macromolecular complex. And I got 4 examples on this slide. A traditional enzyme in red, the drugs are always going to be in red, binds in the active site, and it competes out the substrate, inhibits the enzyme. Here we have a protein-protein interaction inhibitor, where again, the drug binds in the protein interaction interface and competes out the interaction. A third example, very popular in the industry and in the market, you have receptor-ligand interactions, and the drug binds in the ligand binding site and therefore shuts off signaling by the receptor. Can also be the opposite, could also be an agonist of the receptor. And there's another way you can do this. Instead of binding the receptor, the drug could be an antibody. It binds the ligand, titrates it out, and again, inhibits signaling through that axis. These are all K-on inhibitors. I'm talking about the exact opposite, drugs that would block K-off. Now, you could think of multiple different classes of K-off inhibitors. You could think of drugs that would bind in an interface, and this is exactly what auxin does, to give you a little bit more surface area, filling a cavity in the interface. You could think of binding right next to the interface. So the interface is there, you extend the interface a little bit, giving a little bit more surface area. Or you could even imagine drugs where they bind distal to the interface. They're more like a staple, right? They're binding what could be quasi-irrelevant areas of the interacting species, but by doing so, just staple them together. And the important point is that this is not limited. It could occur inside of cells, outside of cells, and it could be pretty much any modality. It could be a small molecule, a complex synthetic, a biologic, or a hybrid modality. So, the options are really largely unlimited. Now, in the review that just came out, we had a table of 50 examples, like, trolling through the literature. I was able to find 50 examples of molecules that meet the definition we put forward of a LOCKTAC molecule that we propose is working through inhibiting K-off. And don't worry about this table. If you're interested, you can go read the review. There will be no quizzes. What I'd like to now emphasize are different consequences of having a LOCKTAC. Now, it's human nature to think that if two things interact to do something and you stabilize that interaction, you're going to have agonism. You're going to increase the output of your target. And in fact, that's true in some instances. And I'm just going to emphasize one here because I think they're all cool, but this one is I think potentially very general, where Lilly had made this molecule that's an agonist of the GLP-1 receptor. This is the same GLP-1 that's in Ozempic and Wegovy and Zepbound, et cetera. Okay. Now, you have to understand GLP-1 is made as a 36 amino acid protein. The first 6 amino acids are cut off. The active species starts at amino acid 7. With a half-life of about 2 minutes, that is cleaved by dipeptidylaminopeptidase 4 to remove the 2 N-terminal amino acids. And then that molecule is about 1,000-fold less active on the receptor. It's essentially dead. And so the molecule, because the half-life of the native GLP-1 is so short, the molecule that predominates in circulation is the cleaved form. Now, that cleaved form, Lilly figured it doesn't bind well because there's a cavity now in the ligand binding site because those 2 amino acids that would occupy the cavity are missing. So they made a molecule, this red oval down here, that would fill that cavity and mimic those 2 amino acids, contacting the receptor and contacting GLP-1. And, that molecule, that molecule essentially restores signaling to the cleaved form of GLP-1. Now, what I think is really exciting about this as a more general example is these kind of molecules, if you think of it, if you just made an agonist for a receptor, that agonist is going to agonize the receptor everywhere in the body that the drug gets to, all of the time that the drug is present. So everywhere, all the time, which you could imagine in many cases is probably not a good thing and could lead to on-target off-tissue toxicity. However, in this example, your drug is limited to enhancing signaling at the time and place that the natural ligand is present, right? So you get temporal and spatial regulation of enhanced signaling, of agonism of your receptor. So this, I think, is a very general principle that can be applied in many different signaling pathways. Now, what I think is particularly interesting about LOCKTACs is even though, it would be it would seem intuitive to think that they're going to activate a pathway, they can also inhibit a pathway. And I'm not going to talk about this example. It's in Amgen's pipeline, very interesting example. But I'm going to talk about kinesin. This example is very intuitive because if you think of kinesin, it walks along a microtubule. Well, every time it puts its foot down on a microtubule, that's like an on rate, right? It's binding the microtubule. Meanwhile, to continue walking, the back foot has to lift up off the microtubule and swing in front of the front foot. That lifting off of the microtubule, that's an off rate. So we had made a drug, this has now been out licensed to Volastra, civilnicib, that essentially glues the kinesin down on the microtubule. So now the trailing foot could no longer lift up. So the kinesin is frozen on the microtubule. And that just emphasizes the point that in a lot of biology, it's essential for the interaction to be dynamic, because if it's not dynamic, the whole system gets gummed up. And so in many cases, a LOCKTAC will be an antagonist. Now, a case I find particularly interesting is transcription. So this is a paper from Michael Erb and Benjamin Cravat at Scripps, where they were going after a transcription factor inhibitor, inhibitor of FOXA1. They had a reporter gene, and they found a molecule that formed a covalent bond to FOXA1 that shut down expression of the reporter gene. Now, when they characterized that inhibitor, they found something interesting. So FOXA1 recognizes a 7 base pair consensus, GTA, AA, CA. However, in addition, there's 3 immediate upstream bases denoted by X's here where there's a weak consensus with some redundancy built in. Now, it turns out that weak consensus really matters, that the sites where FOXA1 is active in inducing gene expression conform to that 5' triplet consensus, whereas ones that are not active do not. What they found is that their drug, WX0223, covalently binds the part of FOXA1 that normally sits right over that XXX trinucleotide, a weak consensus. And when it binds there, it essentially eliminates any preference because it itself provides binding energy. That's the hypothesis. And so now it doesn't matter what your triplet sequence is. All sequences with the 7 base consensus bind with equal affinity. And so they looked across an entire chromosome, and this is just a cartoon of their findings, where in yellow you'd have the molecule doesn't change the occupancy on DNA. In red, you only bind those sites when the inhibitor is present. In green, you no longer bind those sites when the inhibitor is present. So what you could see, and the effect here is pretty subtle, is it causes a redistribution of the transcription factor away from some of its sites towards some novel sites. And it just so happens their reference, their reporter gene, was one of these green peaks, and that's why it's shut down. But this brings up a more general interesting point. And this comes out of single molecule tracking. So if you think of a transcription factor, it has a job to do, right? It has to recognize its sequence in a huge genome. Now for MYC, CAC-GTG is the site. You can calculate that there's 1.5 million of those in the 6 billion diploid genome, yet there's only 33,000 copies of MYC in a cell. So 50 times more canonical sites than molecules. And it's even worse than that because you can calculate there's 18 different one base pair mismatches to the CAC-GTG, many of which are likely to have some affinity, diminished, but some affinity for MYC. And so now you have to multiply this mismatch by almost 20. So there's literally almost about 1,000-fold excess of potential binding sites, and potentially more if some 2 base pair mismatches can bind, et cetera, et cetera. So, it used to be that people thought of transcription factors as essentially having 2 binding characteristics on DNA. You'd have very short binding to non-specific sites or long binding to specific sites. However, when people have measured this by SMT, and here's data for 3 different transcription factors, the survival time on DNA, the dwell time, is actually, it fits a power law. It doesn't fit this bimodal distribution. And you could think of that as there being a lot of different sites of variable affinity, because again, mutant sites will have some degree of affinity. Upstream and downstream nucleotides will affect affinity. Other proteins bound in the area will affect affinity. And so a transcription factor, to do its thing, has to find its critical sites in front of its real target genes. But it doesn't have a GPS. It can only do it by trial and error. So it's going to have to bind a lot of times in sites that are not active for it to find the sites where it has to do its job. And that then creates this counterintuitive idea that perhaps the best way to inhibit a transcription factor is not by inhibiting binding to DNA, but by stabilizing it and blocking the search. So I'll just quickly go through advantages of LOCKTACs over conventional competitive inhibitors. The substrate and binding site of a ligand site of a conventional inhibitor tend to be highly conserved. Adjacent regions where you could bind a LOCKTAC are not. And so that opens up for specificity. The natural ligands that bind sites that competitive inhibitors bind can be very abundant and have high affinity. So for example, with RAS, GTP is at 1 millimolar and has picomolar affinity. Overcoming that's pretty hard. Number 3, with a LOCKTAC, the template interface that you want to target pre-exists in the cell. Whereas for a molecular glue, for example, that you're making an induced proximity molecule, you have to create a neomorphic association, which just feels intrinsically much more difficult than going after a templated site. Number 4, I would just submit as a biologist that if you can reduce K off, of a relevant interaction, it's got a high K-off for a reason. You're almost certainly going to have an impact on your target. Whereas proximity inducers, you can make a ternary complex. You can bring a horse to water, but you can't make a drink, right? In many cases, you don't get ubiquitination with degraders, for example. Finally, if you want to block a protein-protein interaction, you have to overcome a lot of surface area, potentially 10 to 20 kcal of binding energy. With a LOCKTAC, literally, if you had a single hydrogen bond, plugging that into the Gibbs equation, you could potentially reduce K-off by up to 150-fold, which would be like taking a 1-minute half-life and turning it into 2.5 hours. And so, again, as a biologist, I submit that's almost certainly going to have a significant consequence. So just to conclude, and I'll be happy to take questions, there's a great need for innovation in drug discovery. Multispecifics and proximity-based therapeutics are really redefining druggability and opening up the potential of what you can achieve with pharmacotherapies. Now, many induced proximity drugs, particularly degraders, have entered the clinic, and we could expect to see many approvals in the coming decade. And there's increasingly novel approaches to multispecificity and induced proximity. I outlined sustained proximity as one example, but there are others. And so I think the future is bright. For those of you who want to go into drug development as a career, or maybe as I did as a second career, I think there's enormous potential to make really impactful molecules that will transform human health. And so with that, I thank you for your attention. Be happy to take any questions.
Thank you so much, Ray, for a wonderful presentation, inspiring work. I do have a quick question myself, is how hard do you predict discovering LOCKTACs are going to be compared to discovering molecular glues in the future, as well as proximity, induced proximity drugs?
Well, it's funny you should ask that, because I got an email yesterday from Jim Inglese, who I might be on this call. He said he was going to try to make it. And he just published a paper on a method that would enable identifying molecules that affect the abundance of ternary complexes inside of cells, right? So you're correct in noting that directly monitoring K-off would actually be quite difficult. And even monitoring steady state level of a complex in a cell could be difficult. But Inglese has found that, using an alpha complementation approach, they could see when two proteins come together, you have some change in the alpha complementation that occurs with, the high bit N-LUC fragments. And you see a change in activity, and that is quantitative and very easily measurable in high throughput. So I think, there's going to be other methods, but I think it's clear that there's emerging methods that can be applied to seek directly molecules that change the steady state abundance of complexes through reducing K-off.
Fantastic. Thanks, Ray. So we do have multiple questions, so I'll go through them, and everyone can post new questions as well. All right, so here's one. Could you address potential rise of resistance or tolerance to multispecific drugs in case of infectious diseases?
Yeah, this is a question that always comes up, and there's always the possibility of resistance. What's interesting, and some might think, well, resistance would be a bigger problem because now you have two things that are involved in the cell. So maybe you double the resistance problem. Despite that, lenalidomide Revlimid has been frontline mainstay in multiple myeloma therapy for the past 20 years. And so, resistance is an issue. It's not unique to these drugs, but I would say the example of Revlimid, cyclosporin is another example. These drugs show that it's not an insurmountable obstacle. But it is always something you'll have to worry about, whether you're a multispecific or a monospecific.
Fantastic. Thank you, Ray. All right, so here's another question.
Well, let me add one addition to that. The multi-active category of multi-specific potentially is more less prone to that, right? Because you're addressing multiple nodes at the same time with a single molecule.
Makes sense. All right, so here's another one. Regarding molecular glues for broad utility, what strategies may be needed to discern between false positive hits from the mountain of needles and ones that could be targeted for specific outcomes?
I'm not exactly sure what the question is getting at. So, false positive hits are always an issue. I think there are clear ways to sort through that. One thing we know with lenalidomide that's been a problem is that it's recognizing a particular beta turn motif, right? So it's been shown by Nico Toma and Ben Ebert that I think there's literally like 300 helix loop helix transcription factors that show some degree of targeting by lenalidomide. And that almost certainly accounts for the birth defects that were caused by thalidomide, which is in the same family. So in that case, there's a way to monitor your activity against those other targets. You can use, what Ebert and Toma did, use proteomics approaches to find those. I think it's an open question as to whether, because lenalidomide is recognizing a particular structural motif that's fairly redundant in transcription factors. Different glues are going to recognize different types of motifs. And so I would expect this is not going to be generally true of all glues. And, it's probably going to be very dependent on the particular ligase that's being employed, for making a glue like drug. I'm not sure if that addresses the question, but I hope so.
Oh, thank you. And if they have a follow-up question, feel free just to add it to the chat. All right, so we'll move on to the next question. The proximity field has adopted new lexicon like linkerology. What about complexology, which I invent the definition of here as the study of the order of formation of a ternary or higher complex? It's a long question, Ray. Bear with me. Let me read it through it. I think we are gathering an understanding of E ligases. Remind us if these tend to form weighted in equilibrium over target of interest binary complex, or does it vary a lot? But regardless, other proximity systems will define by target, by expression locations, other complexologies. So I guess this is the question. Is this a field worth pedagogically understanding better, or do we just keep trying proximity designs in a case by case, or take what we get stochastic approach? Does complexology matter in the proximity design process? I'm not sure if you got that.
I think it does. And, one of the companies that really invested effort in this, and now I'm going to forget the name of there's an investigator at Yale in the chemistry department, whose name I'm not recalling at the moment, who developed mathematical methods to estimate ternary complex formation as a function of the affinity of an inducing agent with the different surfaces that it's contacting. And what came out of that is some sort of non-intuitive findings in terms of how you want to dial in the affinity for the different components. Because what you want to do is, you don't want to form the two binary complexes. You just want to form the ternary complex, because the binary complex, will work at cross purposes, not going to get the job done. And so, that required a pretty sophisticated understanding of macromolecular interactions to develop that model. And so, Chimera has used this in their design and development of degrader molecules. And, I think they've been one of the leading companies in terms of their success to date. And so I do think that understanding of biophysics of how things are coming together is going to be important. And yeah, you can do it, just empirically, and that's been done. But, to take it to another level, I think figuring out, more systematic rules that are rooted in biophysics will be important.
Fantastic. Thank you. So there is more questions here. Do you mind staying on a little longer so we can take
I'm
some questions?
totally fine with that.
All right, I appreciate it.
Dan, as long as
All
you
right.
want.
Oh, fantastic. That sounds awesome, but we won't take too much of your time, I hope. There is a question here that has a lot of likes, so let's take that one. Wondering if there are examples of proximity molecules that prolong the interaction of GPCRs with G proteins, thereby inhibiting agonist-promoted GDP-GTP exchange.
Well, the example I showed of the Lilly molecule, that's an agonist, right? That's a GPCR. So there's at least an example of an agonist for a GPCR. I can't think off the top of my head if there's an example of an antagonist. If there is, it would be in that table 1 of the science review that came out last Thursday. So I'm sorry, I can't, I just don't recall.
No worries. I think people should probably read that, and I'll read it. So that sounds exciting. All right. So here's another question. PROTACs seem well suited to promoting degradation of proteins that aggregate and cause some neurodegenerative diseases. As PROTACs seem to be relatively large, is their use of neurodegenerative diseases limited due to the need to penetrate the blood-brain barrier?
Yeah, that's going to be a challenge, right? I know that there are companies that are going after, targets that are in the brain using a PROTAC. I believe Arvinas has, publicly disclosed that they're doing that. Getting through the blood-brain barrier is always a challenge for drugs. And so it's potentially a bigger challenge for a PROTAC-like drug if it is higher molecular weight. That said, there have recently been reports of mechanisms that could potentially be applied to small molecules as well for helping them. And there was just a deal announced yesterday. Novartis did a partnership for molecules, for strategies, multi-specificity strategies, usually through transferrin receptor, for helping shepherd molecules across the blood-brain barrier. So it's a challenge, but I do think there are potentially creative solutions to solving the challenge.
Great. How successful was your strategy at Amgen to use DEL to find new tractable chemical space for novel E3 ligases?
We had tremendous success. I think we found, I forget the numbers now, over 300 different ligands for about 24 ubiquitin ligases that we could validate in, sort of manipulated systems that we could validate that were actually working. So that's one approach, but there's a whole other approach, which is just, building libraries that would allow you to look for fortuitous interactions, more glue-like things. So I think we will see more ligases joining the fray besides CRBN and VHL, which have really dominated the conversation so far, but largely for historical reasons. I don't see any reason why this won't be far more general than it currently is.
Awesome. And just a follow up from me now. So when you said there's 500 genes encoding the 3 ligases, and I'm guessing these genes are ubiquitously expressed, like they express depending on different tissues, and so that can also help with specific targets in different areas in the body as well. It's just not being explored yet. like that heavily.
People are thinking about this. I'm not aware of any drug that's in clinical development yet that exploits that. When Ryan, the colleague I co-wrote the review with, Ryan Potts at Amgen, when he was an academic at Dallas and then Memphis at St. Jude, he was working on a family of proteins, the MAGE proteins, that are tumor-specific antigens. And it turns out they're subunits of ubiquitin ligase complexes. And so, that could be an example, the MAGE proteins of getting ubiquitin ligase that's tumor selective, right? So, of course, T cells are probably going to have their own unique ubiquitin ligases. So you could imagine, making degraders that might be specific for certain subsets of cells in the immune system. But again, there's been a lot of, I think, armchair talk about that, but no molecules in the clinic yet, at least that I know of.
Thanks, Ray. Okay, lots more questions, so I'll take some more. With LOCKTACs, can cells sense that a normally dynamic reaction is now sustained? Would that trigger a compensatory response? Or would you simply not pursue that LOCKTAC then?
It certainly could trigger a compensatory response, right? We know, for example, that if you lock receptor signaling on, that there's especially with GPCRs, there could be a compensatory response of receptor downregulation. And so, yes, there may be compensatory responses. Whether or not you would pursue that, there's a lot of GPCR agonists out there. GLP-1, obvious example. So even though there are potential compensatory responses, it doesn't vitiate the potential utility of drugging the target. And I think, that's just going to have to be done on a case-by-case basis, right? Because in many cases, we won't even know unless somebody's attempted to do it before if there's a compensatory response until we make the molecule. And then, that response will have to be studied to determine, whether it is such that it essentially precludes development of the drug or not.
Yep, makes sense. Here's an interesting one. Is a LOCKTAC an invitation to off-target effects?
Is a LOCKTAC what?
An invitation to off-target effects. I guess they're worried about all the off-target effects that you might get out of the LOCKTAC.
I guess my hope, and I laid that out in the next to last slide, is that you might have less of an off-target effect because unlike a classic competitive inhibitor, you don't have to target the region of a protein that's highly conserved, right? Because usually a ligand binding site or an active site are highly conserved. And so by not having to target a highly conserved region, perhaps you might end up with molecules that have a lower tendency to have off-target effects. Now, the other thing is the LOCKTAC, I had the one example of an agonist of receptor where it bound a ligand and it bound a receptor and just held them together more. That could be an antibody. And then when you're talking about antibody-based drugs, then, off-target effects becomes much, much lower concern because you can develop molecules with, picomolar affinity. So, I'm optimistic that you could reduce that problem with the LOCKTAC mechanism.
Thanks, Ray. We'll take a question. This is from Jim Inglese, actually. So let's see what he has. Wonderful talk, he's saying. How prevalent do you think nature's use of small molecules, example, auxin, etc., are in using targeted proximity for defense and signaling? Should we be looking at natural products in a different way than we have traditionally done in the past?
Well, that's an interesting question. We know that plants do it for more than one hormone, right? We know they do it for cytokinins as well. So, they've landed on the same solution at least a couple of times. Viruses, as I outlined, make small proteins frequently that induce proximity between a cellular protein and a ubiquitin ligase. So yeah, I think the idea of using induced proximity to achieve a pharmacological effect or a desired phenotypic effect is commonplace in the natural world. And I think, his point about small molecules, thinking about this from the point of view of small molecules, is an interesting one. Certainly that's the case if you look at rapamycin works through induced proximity. That's a natural small molecule. Cyclosporin works through induced proximity again. That came from a natural source. So, and those were the first molecules ever defined to work through induced proximity. So, I do think that it's actually a really interesting idea to deliberately look for this in natural products. Oh, Taxol is another, although Taxol is complicated because it's not a LOCKTAC in the sense that it's acting allosterically, but there are other molecules that, like Taxol, stabilize microtubules against depolymerization that are directly at the interface between alpha-beta tubulin dimers, and they're directly stabilizing the interface so that they don't depolymerize from each other. And again, Taxol comes from a natural source, right? Pacific yew bark. So yeah, I think that's systematic searches for those type of mechanisms might be productive.
Awesome, thanks, Ray. We are 12 minutes past 2, so I'm going to stop here. There's a lot of other questions, but I would like to thank you for your time here, and maybe we can continue these conversations. We can invite you here to NCATS in the future, and I'd love to continue these conversations with you. It was a fascinating talk. I appreciate you being here, and thank you for your time from all of us here at NCATS and our attendees. Thank you so much.
Great. Thank you so much for the opportunity. It was really my pleasure. It's always such a gift to be able to come and talk to scientists such as yourselves, and talk about the latest things on my mind. And there's nothing like being retired to appreciate that gift even more. So
we're always happy to hear. Yeah, thank you. And everyone, thank you so much for joining us today. Our next webinar is going to be by Dr. Peter Sorger, and it's going to be in Q4. Just follow us. We'll send follow-up updates for everyone. And this webinar is being recorded, and we will send you the recording very soon as well. Thank you, everyone. Thanks, Ray. You have a wonderful day. Bye.